Today we continue our series of seminars on Wednesdays, and it's my pleasure and privilege to present Dr. Jonas Schmidt-Chanasit from Hamburg, Germany. He is a professor at the Bernhard Nocht Institute for Tropical Medicine. He's into three different areas of research. One, he's in charge of the study of arboviruses in the European community. They have already identified three different viruses since 2009. These viruses were not previously present in Germany. He is also an advisor for the World Health Organization, and he's collaborating with the committee, the Olympic Committee, organizing the Olympics in Rio next year. And he is also studying the ecology of Arthropod-Borne Viruses in the Danube River. Today he is presenting the emerging viral diseases, lessons learned from recent arboviruses. Dr. Schmidt.
Yes, ladies and gentlemen, dear colleagues, first I would like to thank Dr. Gerardo and Dr. Carlos Medina for the kind invitation, hospitality. I enjoyed the first 2 days a lot. It's my first time in Mexico, and I'm really impressed of this research institution in UANL. So I hope that we can strengthen our collaboration in the future and will establish some very nice research projects. Yeah, let me start with my presentation. I try to focus on two subjects. One, the arboviruses, and second, the Ebola virus. Within the arboviruses, I will focus on one arbovirus because we are limited in the time, so I will talk about the Zika virus, which is currently also an emerging arbovirus in South America, in Brazil, as you may know. Try. I think the remote will not work. Not work. Ah, yes, now, okay. It's a little bit slow. I think it's not really working. A moment. No, I think it's not working. I have to make it there.
Do not trust technology that much.
If not, I can give you a hand sign and you just will show the next slide. I mean, it's, oh no.
I'm not sure. The good thing is that it's being broadcasted everywhere.
Yes, so this is the real situation in presentations. There will be also always some errors, so let's try if we can fix it. Ah, okay. The USB stick needs to be connected to the computer, otherwise. Yeah, this is a very interesting video, and I would like to talk or tell you some story about this, but step by step, so we need to start with the first slide. I'm sorry. Okay, now it should work. Okay, when we're talking about Arthropod-Borne Viruses, I would like just to mention that, or to make you remember that this is a very, very diverse group, including millions of known species. You know, butterflies, spiders, mosquitoes, bees, and wasps. compared with the other species on Earth like mammals. So just make you remember that this is a really diverse groups that we have to face when we're talking about arboviruses. However, our relationship with these kind of species, let's say, is somehow negative influence. So mostly when we're thinking about insects, I think we're thinking about to control them and not see the benefits. I think only the butterflies and bees have a positive emotion or cause a positive emotion in our minds. Mostly we try to kill insects, and we are the only species on Earth that is able to kill millions of insects within seconds. However, in the past, you know, I just found this very nice newspaper. In the 50s, I think, they treated even the children's wallpapers with DDT. However, this came back to us, you know, the story about DDT. And I was thinking why we have this negative influence thinking about insects. And now I came to this story, and I was thinking that maybe it was seeded in our brains long time ago, millions of years ago. And this is just a short video showing you a story. This is us, our ancestors, Myllocomingia, a fish-like animal. And at this time, there was an arthropod predator, which is called Anomalocaris. It was the largest predator of this time. And during the Cambrian era, this was the only area where arthropods were able to hunt and eat humans, or our ancestors at least. And I think this is just an interesting fact to see the evolution, which is very interesting. So we developed from the fish-like animals to reptiles, to birds, and finally to humans, and now it turns and we are able to kill and trap insects. However, I would like to just show you also one more positive image of insects, and this is just a picture that I took some weeks ago on my balcony. So this is a cactus, Escobaria missouriensis, and you can see here, this visitor is a fly. So not only bees are important for the pollination, there are also flies, even in Germany, together with a Mexican cactus. So it is important that we have this insect, that this Mexican girl will survive and replicate. So just a positive picture for the function and usefulness of insects. This cactus is named after two important pioneers in Mexico for agricultural science. These two brothers, Escobar brothers. So I just like to show you this picture because I'm in Mexico. However, when we're thinking about insect science, when it started, I have to mention one important monograph. This was published by Jan Swammerdam. He was a medical doctor. At this time, biological science was not established, so mostly research was done also by medical doctors, doctores medicinae. And he demonstrated for the first time that the different life stages of the mosquito, so the pupae and the larvae, belong to one individual, not to several species or several individuals. So this was a very important finding, that he demonstrated that this is one individual. Another colleague demonstrated for the first time a causative agent of a disease, and this was the itch mite, which caused scabies. So this, or thus scabies became one of the first diseases with a known cause, and it was caused by an insect. I like to show you this picture. It is from Peter Bruegel the Elder, and it was painted in the 16th century, and it shows the terror that was caused by the Black Death, the plague. And when I watching to this very beautiful painting, I was somehow remembered about the television and pictures that I saw 1 year ago during the Ebola outbreak. Scenes of not terror, but anarchy in the streets of Monrovia. So it really resembles this situation, even not in this epidemic stage. So why I was talking about this, because I would like to tell you that all these pathogens that we're now talking about are zoonotic agents. So they are transmitted from a vertebrate to humans. So this is a direct zoonosis like rabies or Ebola, which we are talking about, or which I will be talking about soon, and plague, or the Black Death, or arboviruses are metazoonoses. So they need a vector that transmit a pathogen from the vertebrate host to humans. I took this picture 3 years ago during a congress in India, and this represents very well the severe situation for the public health and the humans in these hyper-endemic areas for dengue or chikungunya virus, which is highly prevalent, especially in India. So they put these stickers on all windows in the hotel. So we are not allowed to open the windows or the balcony doors because the mosquitoes that are here around, they may transmit dengue and chikungunya or Japanese encephalitis virus. So it's a really severe problem in these areas of the world. And this is also very nicely illustrated by this picture from a recent work in Nature that shows you the hyperendemic areas for dengue. India is a hotspot, Brazil in South America, Mexico as well. In Southeast Asia is Indonesia, Thailand, and Vietnam. And according to this work, the estimates around 400 million cases of dengue every year, of which around 100 manifest apparently. So these are new numbers. The old numbers from WHO are around 50 to 100 million cases. I also like to show you this picture. We have a system in Germany that every imported dengue virus case needs to be reported to the public health authorities in every state, and they will report these numbers to the Ministry of Health. And this is a very, very important thing. First, you can see the development in the cases. So there is a strong increase in the numbers of imported dengue virus cases. And this represents also the worldwide trend. So this is not only important for Germany, it's the same situation worldwide. So we have a strong increase in the numbers of dengue virus infections. But the second thing is that only with this reporting system, we know what's happened and where these patients come from, because mostly we receive samples from all over Germany or even all over Europe, but we don't have any anamnestic information or any patient records. So we don't know where the patient was traveling. And this is demonstrated by this picture. So just imagine that this is our reference center, this spider, and these are our patients, so the mosquitoes. They come from all over the world and they are trapped. And we know very well what's going on a worldwide scale, which virus is circulating in Southeast Asia and Africa, or even in the Central Americas. But this, this benefit is not based on very fancy diagnostic assays. So this is not the main point, that we have very good PCRs or next generation sequencing or very well immunological methods. No, the main important thing is the spider net, and the spider net is the public health system. So the public health system report us where the patient was, where he was traveling, when he came back, and this is the most important information to diagnose these primary cases. For example, the first imported or the first dengue virus case acquired in Japan, or the first Onyongyong virus infection that occurred outside of Africa, or the first Zika virus infection that was imported to Europe. So the diagnostics of these cases was only possible because the public health sector works closely with us, the public health system, and we got the information about the patients that we usually not got with the samples, because we just received the samples with the patient name or even some number, and we don't know anything. So this work is really important. So I'd just like to give you one example for this case. So this was the first dengue virus case that was acquired in Japan since the Second World War. So this German traveler was two weeks in Japan, visited several cities, and she recorded multiple mosquito bites here in Fuifuki. So most likely she became infected here in Fuifuki. We performed the diagnostics, and it was clear that she was infected with dengue virus. So we have a positive NS1 antigen, so direct virus detection method, so antigen, and we were able to serotype this dengue virus as serotype 2. We alerted our Japanese colleagues about these findings, the public health ministry or the health ministry in Japan, and they conducted shortly after this report also a study and analyzed if it might be possible that the patient became also infected in other cities of Japan. What you can see as the red color means that there are suitable conditions that the vector can survive, which is Aedes albopictus. Aedes albopictus is not a new invasive species in Japan. It's present since many decades, and it was the responsible vector species for the large dengue virus outbreaks during the Second World War in Japan. So since the Second World War, there was no autochthonous cases of dengue anymore in Japan. So according to this data, it was also possible that she became infected, for example, in Tokyo, because there are suitable conditions and the vector is present, not only in Fuifuki. Another study by our Japanese colleagues analyzed the possibility if there can be an outbreak. Is there a suitable condition for an outbreak? And what they find out, I don't understand this, but this picture is really clear for me. And what they demonstrated was that even there's some numbers of primary cases, not only one, even if there are 5 or 10, the reproduction number will be below 1, so there will be no outbreak. You know, the reproduction number, for example, during the Ebola outbreak was 1.5 up to 2, for measles it's 9, but they calculated that it will be below 1, so there will be no outbreak. When I'm looking on these calculations and these models, I always thinking about this guy, a very famous statistician. And I always talk to my students, please remember what he said. And he said, essentially, all models are wrong. All models are wrong, but some are useful. And this needs to be proven here with this model. And it was proven exactly 1 year after this first case. There was a large dengue virus outbreak in the city center of Tokyo. Here reported also by CNN. Hundreds of dengue virus cases were diagnosed. And this outbreak was just under control because they closed the park and they performed an extensive mosquito control. So it was, area 0 was not below 1, let's say it like this. And I think this is a nice example for Arbovirus emergence. It can change completely the situation within 1 year. There were 70 years without any circulation of dengue in Japan, and then within 1-2 years we have a severe outbreak. Dengue virus is circulating in Japan, and this is also interesting because this outbreak was caused by dengue serotype 1. Our patient was infected with serotype 2, so there are already 2 serotypes probably circulating in several regions of Japan, and now the Japanese colleagues, for sure, they do a lot of studies to find out where the virus is circulating and in which mosquito species, not only maybe in Aedes albopictus, but maybe other species are involved. Who knows this? Because of this example, we try to be prepared in Germany, and we, Dr. Gerardo already mentioned it, that we initiated a mosquito and mosquito-borne virus surveillance program in 2009. This is just an example of the mosquito monitoring program. So we have these squares all over Germany. This is for a period of 3 years, now from 2015 until 2017. So you have these squares, is 50 to 50 kilometers, and within these squares you have 3 trapping sites. And these trapping sites are randomly chosen according to Korean land cover data. So you have near rural, rural, and urban. And they will be changed every year. So we trap mosquitoes here, we will trap mosquitoes here and here, 2015, 2016, 2017. This is impossible to do with our resources, so we use volunteers that perform these trappings, which are done with BG traps. And with this data, you are able to give a very precise picture about mosquito distribution, about the invasive mosquito species that are already present in Germany. I will just give you 3 examples. This is an invasive mosquito species, Aedes albopictus, which can be found here in the southwestern part of Germany. Interestingly, mostly along the highways, because you know, Italy is heavily infested by these Aedes albopictus. They have severe problems with this invasive species, also in France and Spain. So the trucks came from the border here from Italy, and they import these invasive mosquitoes into Germany, and now these mosquitoes also replicate here in this region. So they have no problem to survive minus 20 degrees or minus 10 degrees. There's no problem for the Asian tiger mosquito. So you can also see that it go more to the north. No? So it started here in 2012, and then you have the green dots here, the first appearance in 2013. So it spread. And you know, it's a competent vector for a lot of different arboviruses, not only dengue, also chikungunya, Usutu West Nile. A very, very competent vector. This is also an interesting picture. So this is Germany, and this is an Anopheles species. And according to the data or the methods that I showed you before with the squares, we're able to model the distribution and the habitats. So what you can see here is, for example, for Anopheles dacia, that it is mainly present in these parts of Germany, which are more warm and have, show low precipitation. So you can't find this species, for example, here in the western and the southern parts of Germany. The same was shown for this species, Anopheles arthroparvus. So we have just ideal conditions here in the very, very far northwestern part of Germany, close to the border to the Netherlands. This is the methodology or the system for the mosquito-borne virus surveillance. So the squares are larger, it's 250 kilometers, and we trap in every square 25,000 mosquito every year. So all over 350,000 mosquitoes every year need to be trapped and to be analyzed and screened for viruses. So this is our mosquito-borne virus surveillance program. So it needs a lot of manpower, a lot of material, but to be sensitive enough, you need to trap these large amount of mosquitoes. Otherwise, you will not detect any virus. If you just analyze 1,000 or 10,000 mosquitoes, is not enough. You need to be very sensitive. You need high numbers of mosquito to be able to detect something. And Dr. Gerardo mentioned it already that in the past, we detected 3 viruses in Germany: the Usutu virus, which is closely related to West Nile virus, Scabies virus, which can also cause human disease, and Bataille virus. All of these viruses are detected here in the southwestern part of Germany because there are ideal conditions, weather conditions for arboviruses. It's very hot here, very humid, and you have the Rhine River, so a lot of flooded areas. Now I would like to talk about the Zika virus, which is also very interesting arboviruses. Arbovirus. I choose this picture from William Hodge. He traveled together with James Cook during the second voyage, and this is in Tahiti. And I was just thinking about how it's possible that these kind of arboviruses can spread over thousands of kilometers. Nowadays, it's clear they can use the airplanes, but also in the past it was possible. Just think about the yellow fever introduction to South America. And here in this painting, you can really, it's nicely illustrated how it's possible. You have all the factors that lead to the survival of the eggs or the larvae. So you have water reservoirs, you have children, you have birds, fruits. So even at this time, it was possible that these kind of viruses travel hundreds of kilometers. And the students often ask me where all these names come from: Dengue, chikungunya, Zika, Mayaro, and I just would like to explain to you for the Zika virus, just one example. So this is the original publication about the Zika virus. So the most productive time in arbovirus research was in the 30s, 40s, 50s, 60s of the last century. There were a lot of arbovirus research stations in the tropics, mostly founded by the Rockefeller Foundation, for yellow fever research. And this was one important here in Entebbe. It still exists, however, not so much focus on arboviruses, but it's still a reference center, mainly for Ebola and Marburg viruses in Uganda. And close to this research station, there was a forest called Zika. And during this time, they tried to isolate new arboviruses in that manner that they take rhesus monkeys in cage, and they put them in the canopy of the trees, and they waited until these monkeys became febrile, and then they take the serum and inject it into the brain of baby mice. So it was possible to isolate a lot of different arboviruses that we have to face now, like West Nile was isolated in the same way with the baby mice. They also conducted a lot of interesting ecological studies in the 50s, and here they identified the vector species for the Zika virus, which is Aedes africanus, so Aedes species. And you can see here the steel tower. This is the Zika forest, and they have the steel tower, and they trap mosquitoes here in the different stages of the forest. So this is 20 feet, and this is 100 feet. And they identified a very interesting ecological feature of this virus. Mostly positive mosquitoes were trapped here in the canopy during the dawn or in the night. So you know in the tropics there's a light wind in the dawn, so this will lead to the passive distribution of the mosquitoes and the virus over large areas, yeah, because normally this mosquito is not able to fly 10 or 20 kilometers, just maybe 100, 200 meters. But with this wind, it is possible that it will drift along away. So very interesting study, even in the early 50s of the last century. So Zika virus was first discovered here in Uganda, also demonstrated in mosquitoes here in Nigeria, in Senegal, and there was serological evidence for the circulation of the Zika virus here also in the Southeast Asia. And in 2009, there was a large outbreak on Yap Island, which belongs to Micronesia, or 2007. But then in the Yap outbreak. But then in 2013, it emerged in French Polynesia, which with around 9,000 cases. And then what I mentioned before, it spread like thousands of kilometers to the west to cause an outbreak here in New Caledonia. Then it spread to the Cook Islands, which belong to New Zealand. We have imported cases to Australia. And then you can see here this arrow. There's not enough space because it's too far. It spread also to the Easter Islands, yeah, in March 2014, and caused also some cases there. And I prepared this picture, I think now one year ago, and I already put this arrow here and asked for the possibility that the virus might be introduced into South America because the competent vector, Aedes albopictus, Aedes aegypti, is present. And now it happened just some months ago that we saw the cases, or that we saw Zika virus cases in Brazil. Even maybe it's a little bit longer circulating, but it was just diagnosed some months ago because, you know, it can be easily taken as dengue virus cases. So what is also typical feature of this arbovirus outbreaks is that you have an explosive spread among the islands, a lot of patients, but then it disappears in the same way. So within weeks, it is not there anymore. So this is a typical feature of arboviruses. So mostly if you recognize that there's an arbovirus outbreak, it is too late. You are already here. Yeah. So this is also a typical feature. The same for chikungunya. This is some summary about the clinical presentation. So mostly it's not easy to distinguish from any other arbovirus infection like dengue. So fever, rash, myalgia, polyarthralgia. Maybe polyarthralgia, not so prominent, but myalgia. So you can see here, even for this one case in Uganda, one case or 7 cases in Indonesia. So this was the outbreak in Yap. And you can see even here sometimes they have headaches, sometimes not, sometimes they present with rash, sometimes not. And so it is not very clear. However, we can say sometimes compared with dengue, you often see more conjunctivitis, but okay, you need to analyze more patients and to observe more patient data to really say this. However, I just marked this in red because this is really interesting. Here you can see the United States. Zika virus is not circulating in the United States, but this was a case, a biologist, he trapped mosquitoes in Senegal, and he acquired the infections in Senegal, traveled back to the US, and infected his wife during sexual intercourse. And this was the first documented case that the arbovirus was transmitted was the first sexual transmission. Yeah, because he collected his semen, he collected the blood, and then he published the case by himself. So really interesting as a first author, yeah, for sure, the first author. Yeah, but you might think, okay, this was demonstrated for dengue or West Nile. No, it was this very rare Zika virus at this time that this sexual transmission was first demonstrated. That it is not only how to say, infection that went within 1 week, so not a severe infection, was contrasted by this data. So they first observed in French Polynesia that there was an association with severe neurological complication, especially with Guillain-Barré syndrome. And this is really interesting because usually you say, okay, the Zika virus, within 1 week you have a little bit fever and then it's okay, it's not that severe. But it was contrasted by this data. And the question is why you have this accumulation of severe neurological complication. Maybe because dengue virus was causing outbreak at the same time, chikungunya virus is circulating, or is the genetic background from the Polynesian. Nobody knows this. But we observe now the same problem in Brazil. So there's an increase in the numbers of severe neurological complications, especially of Guillain-Barré syndrome. So maybe there is an interaction between dengue virus and Zika virus. Nobody knows, but this is a very interesting field of research, and we have to be careful about this because this might be really dangerous in the future. What about Zika virus diagnostics? Zika virus is a flavivirus, so the same like West Nile dengue. There's an extensive serological cross-reactivity between the different flaviviruses. So for example, if you have an isolated IgM for dengue virus, no IgG, no NS1, PCR negative, it's unlikely that it's dengue, but it might be another flavivirus. So this is the case here, which was demonstrated, for example, during the YAP outbreak. So they have isolated IgM results, and they came out as Zika virus infections because of the serological cross-reactivity. What is also an interesting feature is the low viral load in Zika virus patients. Compared to dengue, in dengue you have 1 million, 10 million. Yeah? The viral load is much higher in dengue. In Zika, it's much lower. It's an interesting feature because when you think about the Easter Island, how the virus may survive, because the viral load actually is too low to infect an uninfected mosquito, should be around 1 million, yeah, to lead to an effective transmission to the mosquito and to another patient. Yeah? So it's very low. These are the cases that are diagnosed and published in our reference center so far. So you can see this was the first one imported into Europe from Thailand. So a lot of dengue cases, diagnosed dengue cases, might be Zika in Thailand. This is Tahiti. We have 3 cases. You have the clinical findings, what I told you, mostly fever, polyarthralgia, rash, yeah, sometimes the conjunctivitis. Yeah. But all of them are not severe infections. 2 patients we were able to detect the virus itself, but mostly the diagnosis was based on serological analysis. So virus neutralization assay, which is the gold standard in flavivirus serology and immunofluorescence. What we can do with all these samples, we did some immunological analysis, for example, cytokine levels. So we collected acute and reconvalescent samples and compared this with blood donors, and what you can see as a polyfunctional response, which is comparable also with the response in dengue virus patients. This is the same here for Rantes and IP10, and also for the reconvalescence patient, we are able that the recovery from Zika virus infections associated with the restoration of normal numbers of immune cells. So this is, for example, the beat uptick in dendritic cells. So this is also the first immunological data on Zika virus infection, because so far there were not much cases diagnosed and analyzed in this way. So this is also the first data. Let's change to another subject, direct zoonosis, such as rabies, but now we're talking about Ebola, and that's why I would like to show you this very nice animal, which is a fruit bat. This is not in Africa, this is in Vietnam. So this is a highway in the rainforest, and there is a place where you can feed these fruit bats with bananas. Yeah? And this is how it happens. Yeah? We are too close to these normally protected habitats that are protected from humans. Yeah? But here it come together, and then for sure these viruses that are present in the fruit bats can jump to humans and cause disease. And this is what is the story for Ebola. So normally it's circulating in an enzootic cycle between the fruit bats, yeah, and some animals that live in the forest, for example here, the primates or the antelopes. And sometimes it happens that, for example, the human will become in contact with the fruit bats because they eat the fruit bats, or the fruit bats contaminated some fruits with their saliva or their urine. So this is the way that humans can be infected with the Ebola virus. And then, after this happens, the Ebola virus spread from human to humans. This is the normal way it happens during the epidemic. So there's only, this happened one time, and all the other cases are caused by human-to-human transmission. The Ebola virus, usually you might think is just circulating here in sub-Saharan Africa, but this is not true. It is circulating also in Southeast Asia, and many people don't know this. However, the species of Ebola virus that is circulating in Southeast Asia is not human pathogenic. It's the Western Ebola virus type. However, there's a potential that also maybe in the future pathogenic Ebola virus species might emerge in this region, because it's a potential habitat. In the past, we observed several outbreaks. So the first one in 1976 in the DRC, so that's why the Ebola is a river, so the virus got its name from this river, 1976 here. We have 5 species. The most important one is the Ebola Zaire species, which caused the outbreaks here in Zaire, in Gabon. Then we have the Sudan species here, green, which caused outbreaks here in South Sudan. Then we have the Bundibuyu, which caused one outbreak here in Uganda. And we have the Côte d'Ivoire or Tai forest Ebola virus species, which caused illness in one patient. This was a Swiss scientist, and she dissected monkeys, and she became infected with this work. Normally the case fertility ratio here for these 2 strains is around 90%. For Bundibuyu it's a little bit better, around 50%. And Tai forest, okay, she survived, so it seems to be 0. And Reston is not pathogenic for humans. Another model from 2014, I think this was in September, published in September 2014, so during the peak of the outbreak. And they calculated that according when the help will arrive in West Africa, there will be 3,000, 10,000, or 25,000 cases per day. So very alarming prediction. You remember this guy? What happened was this. We have very low numbers nowadays, only 2 to 3 cases per week. However, please take a more detailed look into this data. So you have here green Sierra Leone. Okay, this is the peak and it disappeared. Liberia, yellow. So you have the peak here and it disappeared. But look for Guinea. So there was not a real peak here. It always oscillates up and down, and this is still ongoing. So the outbreak started in Guinea, you know this, no? And it's still circulating there. So we have to be careful to say the outbreak will be finished very soon. Yeah? The situation for Guinea is still the same. It goes up and down, even at a lower level now. However, it's still circulating. What are the main factors for the Ebola virus emergence in West Africa? One of the main factors is the mobility of the people there. So this is an interesting study, just recently published, and every small dot is a village. And you can see the relationship between these villages. So, for example, in this village, the people married to another province, so very, very far away. They not choose a wife or man which is close here. No, it's hundreds of kilometers away. The same for work. So they not work in this area. They go to Kenema or even to Liberia or to Guinea. So this was the main problem why the Ebola virus spread so far. And this high mobility, so the borders are not very tight, so you just can cross the border. There was no control, and you can travel within one day from one capital from Monrovia to Conakry or from Freetown to Monrovia. So this was the main problem, the main factor. In contrast to the other outbreaks that I show you in Democratic Republic of Congo, and this happened also here in 2014. There was in parallel an Ebola virus outbreak, however, in a very, very remote area. So there was no direct street, for example, to Kinshasa. And this outbreak was under control within some weeks, yeah, completely controlled. And this actually is also the problem that WHO faced. So nobody was aware that in West Africa there will be that problem with this Ebola virus outbreak, because in the past it was always controlled within several months. Yeah. Now this is the situation with the new data from August. So we have still 2-3 cases here, Conakry, Sierra Leone. So in Liberia there are no new cases. You can see it also here. It's a little bit too much light, but so it's around 5 days for the last report, and here's 25 and 30 days. So normally a region or country is declared Ebola-free if there are 42 days after the last case occurred. So this is 2 times the incubation period, which is thought to be 21 days for Ebola virus. These are the laboratory capacities in the region. So we have 3 mobile labs from our institutes in the ground, mostly here in Guinea, Conakry, and Sierra Leone. So one of the most important tasks of WHO is to bring this laboratory resources to the few cases that are still in this area. So you need some kind of mobility, which is often not so easy because the laboratory equipment cannot be transported over many hundreds of kilometers so easy, so it will take some time. So this was one of the main tasks of WHO and the countries to organize, to bring the capacities to the areas where these Ebola virus cases occurs. Yeah, and one problem is that, for example, sometimes a new Ebola case plop up just here in this remote area and you don't have any capacities there to diagnose or to treat the patient. So this is one of the problems that we still have to face. When we're thinking about this estimation, about 25,000 deaths a day, okay, it not happened, but however, but there were some side effects, and one important side effect is there was a complete collapse of the health system, and you can't treat the normal malaria patient in the way that we are normally treated. So you have to add these deaths also to the Ebola deaths. So there is some connection between this. So you have here the numbers of the Ebola cases, so it drops dramatically, but even there are no cases or only 2 or 3 cases. You have a lot of malaria deaths that normally not occur in these countries, yeah, in this extent. So you have to summarize all these deaths, and then finally you will have much more deaths that are related somehow to this Ebola virus outbreak. What we learned again, or what was shown again, okay, Ebola virus is not transmitted by aerosol. So you have to distinguish between droplets. So the risk is around 1 meter and fomite. So when Dr. Carlos is an Ebola patient, I'm safe. I can talk here. He's infected. I'm not in danger. So this is what we learned. And this was also very important for the control, for example, in the airplane and so on. What was also an important question, is Ebola virus transmitted by semen in reconvalescence patients? This discussion is still ongoing. However, the data that we have and the epidemiology of the outbreak demonstrate that it is not an important feature, that it is not transmitted by reconvalescent patient, by male to female via sexual intercourse. Maybe it's happened 1 in 1 million, but it is not the regular way that the Ebola virus is transmitted. Yeah, this is what we learned. And what you also have to know, okay, it's not enough to perform a RT-PCR of the semen samples. This not tell you that it is infectious. You have to do the virus isolation. Yeah? And then you can say, okay, it's an infectious Ebola virus particle that you detected with the RT-PCR. About the symptoms and signs, this was known before the outbreak, especially in the early phase of infection. There are non-specific symptoms. Yeah? Myalgia, headache, chills, fever, neck And this led to, to the adoption that we not say Ebola hemorrhagic fever, we say Ebola virus disease, because especially in the early phase of infection, there is no hemorrhagic fever. There's no hemorrhagic fever. Mostly it's abdominal pain and so on. So you have to be very careful to say or to exclude something if there's no hemorrhagic presentation. It's sometimes in the late phase of the disease. Yeah? That's why we use the term Ebola virus disease nowadays. And this was also demonstrated during the current outbreak. So they say here, hemorrhage are not the main symptoms, according to this data recently published also in the New England Journal of Medicine. What we also demonstrated was that, that fatal outcomes are also the same for other virus infections, but here it was also shown that the fatal outcome is mostly associated with high viral loads. And this leads directly to the question, how is the pathogenesis of Ebola virus infection? So there are several models. One model is that the Ebola virus particles are not properly identified. They suppress the immune system, so they can replicate very well even in immune cells, and this led to the cytokine storm. And this will destroy your body compartments or even the endothel and led to the vascular leakage syndrome, for example. Therapy, there are some experimental therapies available and they were used. I think everybody knows that ZMapp was used, for example, in these two American patients, the nurse and the doctor. We have a post-exposure prophylaxis, which is based on the Ebola virus vaccine, RVSV, vesicular stomatitis virus. This was used in our institute. We have one needle stick injury in 2009, and we used this vaccine actually as a post-exposure prophylaxis, and it worked. The colleague survived. We have antiviral therapy, favipiravir, which was originally developed as an influenza drug. It inhibits the polymerase of the Ebola virus, and we have other drugs available. However, we don't have too much time, so I just like to focus on the vaccine. We have two vaccine candidates. One that was also developed in Germany, in Marburg, and further developed in Canada is the VSV vaccine. So vesicular stomatitis virus is the backbone, and we have a gene of Ebola virus, the glycoprotein cloned into this backbone. So it's the same like the dengue virus vaccine. You use a yellow fever virus backbone and you clone some genes of interest into this virus. Yeah? So vesicular stomatitis is actually not a human pathogen. It caused some animal disease in the Americas. And it's a rhabdovirus, so somehow related to rabies. Interesting. This is the second candidate. It is also using adenovirus, which is circulating in monkeys, I guess, and they also cloned the Ebola GP into this virus. I just like to show you the data for the vesicular stomatitis virus-based vaccine. So this was the study also performed here in Hamburg for the safety, and it was shown that there are no severe adverse events. So it's very well tolerated, the vaccine, and caused very nice immunity. And now, recently, there was a second study published in The Lancet some days ago. It shows that there's 100% protection, which is very promising, and we hope with the use of this vaccine, we can end the Ebola virus outbreak very soon. Let's hope. Okay, I just like to show you some pictures, more relaxing, not so many texts. This is our mobile laboratory. So this is in March 2014. So we started here. So all the equipment is packed in these orange boxes, which are very safe. So if the airplane crashed, these boxes will survive, even the team not, but you still can use the lab, which is important. So we flew here to Conakry, then used the street to Gekedu. This was the beginning of the outbreak here in this region of Guinea, in Gekedu. And we started to set up the lab close to a treatment center with Physicians Without Borders. So this was the first lab. So very easy, very hot, 50 degrees during the daytime, so no air condition. So this is the unpacking of the bags. This is the lab. What you can see here, the sample arrives. This is how a sample is discarded after the testing. And some people ask, well, why our technician is very well protected, but this poor African boy, he's not protected. Why? Is this some kind of discrimination? No, this is a survivor of Ebola virus. He survived Ebola virus, so he don't need to be protected anymore because there's a long-lasting immunity. This is also an interesting picture because the people ask me why they don't have to protect, they're going to test Ebola virus samples. So the trick is this glove box. So you inactivate the sample within this glove box. So there's no virus that can infect anybody. You only test for RNA. Yeah? So do you inactivate the virus here in this glove box, then you can bring the sample out, do the centrifugation, the PCR within this surrounding. So even you can perform Ebola virus diagnostic here if you just have this little plastic box. So you don't need more than this little plastic box, and then you can diagnose any viral hemorrhagic fever agent, BSL-4 class. So you just need this very fancy BSL-4 laboratories that we also have in our institute, or which in CDC, if you like to work with, to culture the virus in cell culture or do some basic research experiments. But for diagnostics, you don't need a BSL-4. You just need this little plastic box for $2,000. So, yeah. There's under pressure. There's under pressure, sure. No? Okay, this was the first phylogenetic analysis. So we received the first samples and we were able to demonstrate that the outbreak-causing strain belongs to the Zaire Ebola virus and that it is not closely related to the strains that circulating for a long time in Congo, for example, in the DRC. So that means this strain was already circulating for some years or even some decades in West Africa. So it was not currently introduced. Yeah, this is very important information, and this is also supported by the fact that you have seroprevalence in the people over there, sometimes 6%. Yeah? So this shows you that even in the past there were some contact with Ebola virus in that region. However, it not led to an outbreak because it stopped by itself because the village was really remote, so it not spread to the main capitals. This is one performance, showing the performance of one lab, the one that was established in Gekedu. So we tested more than 6,000 Ebola virus samples. You can see nearly 50% of all the tested samples are positive. So one team worked there for around 4 weeks, and then you have another team, and one team consists of one physician, one MD, and 3 technicians. So they work 4 weeks, and then they go back to Germany, and another team follows. So it's around 20 teams now. So this was here, I think, in January. What we can do with the samples, for sure, what was also shown in the previous studies, we observed that in the fatal cases you have a higher viral load. We can do clinical chemistry, for example. What was shown is that in 50% of the cases we observed these elevated CRP, C-reactive protein levels, which is interesting, which you not should expect in a viral infection, but was also demonstrated in this patient that was treated in Hamburg. One patient was treated in Hamburg, here the CRP, yeah, and this is the interesting fact. You have the viral RNA decreasing, so there is no virus present anymore, but this was the critical phase, yeah. So the bacteria infections, the sepsis, this was the critical point. When the patient was not in the ICU in Germany, for sure, he will not survive this infection. Not because of Ebola, because of the sepsis. Yeah? This was the main cause. So you can see here the antibiotic treatments, the CRP, virus not present anymore, but this was the main problem. What was also observed with this patient in Hamburg was also a very interesting feature. Again, the virus load in the plasma decreased, so there's no virus present anymore in the blood. However, we are able to detect the virus here in the sweat for a long time. And even when the patient was released, he was positive for Ebola in the sweat, yeah, in the RT-PCR. RNA, not isolation. In the urine, it was also positive, and here I marked it. This was the last day that virus was isolated from the urine samples. Yeah? So even it was not present in the plasma anymore, it was present in the urine and was still infectious here. Yeah? And then it also disappeared and was only present in the sweat. This is also a very interesting phenomenon, what we can't explain, how this works. There must be some cells that that are not attacked by the immune system, so it still replicates somewhere. Yeah. However, it is not infectious in the sweat, so it's a really interesting thing, and it was observed also in many other Ebola virus patients, not only in this Hamburg patients. Yeah, that you can detect Ebola virus RNA in the sweat for a long time, 40, 50, 1, 2, 3 months. Yeah, that you are able to detect virus in urine. Even they are not anymore circulating in plasma, it is a prominent feature also for arboviruses, for West Nile, for dengue, also for Crimean Congo hemorrhagic fever virus. Yeah? You are able to detect them in urine for a longer time compared to the detection here in plasma. Okay, now I'm finished, exactly 1 hour, and I would like to finish this talk with this very nice picture. So this is one inclusion in amber. And this is really old amber. You have the Dominican amber from the Dominican Republic, for example, which is around 20 million years ago, but this is much older. This is from Cretaceous age. This is 120 million years ago, and this is a biting midge. It still exists nowadays and nearly looked unchanged from a morphological point of view. Not many change. So it just show you how successful these insects are. The same time, we look like this. This is our ancestor, little rat-like animals jumping around. Yeah. So you can imagine how successful these insects are. And I'm always thinking what happened during this time where always something transmitted between us and these blood-sucking insects at this time. And it will be really, or would be really interesting to analyze these kind of insects, to sequence something, yeah, and we are going this way. I mean, we're able to sequence the human genome that is 1 million years ago from Homo neanderthalensis, for example, yeah. So we try to sequence more and more. However, this is really 120 million years is a hard task, I would guess. But with the new techniques, next generation sequencing, however, I think we can do this and adjust our molecular clocks for these phylogenetic analyses in a more precise way. That's why this is really interesting. Okay, thank you very much for your attention.
We thank Dr. Schmidt-Chanasit, and we will wait for the microphones since this seminar is being transmitted through internet. Los micrófonos, por favor. Now, I will request of you to kindly make questions very concise and very brief. Dr. Ramos.
Oh, thank you. Very nice talk. Could you describe the mosquito control in Germany?
Yes.
And if there is data of resistance of the mosquitoes to the chemicals to use to control them?
Yes. The mosquito control in Germany is performed by the German Mosquito Control Agency, which was established 30 years ago in the late 70s. They started to control mosquito in a biological way, so not with chemical compounds. So they use one toxin, which is produced by Bacillus thuringiensis israelensis. So this is a larvicide. So they go into the fields like starting in March and April, looking for the mosquito population, looking for the flooding, and according to this data, they treat the water or the habitats with this toxin, and it's very effective. And until now, there were no resistance for Bacillus thuringiensis israelensis. They're also using helicopters to reach more remote areas. However, I have to mention that this method of mosquito control is a very expensive one. So they ask money from all the cities in this southwestern part of Germany that are normally very hardly hit by the mosquitoes. So they cannot do any business because there were thousands of mosquitoes even during day. So they have a strong interest that these mosquitoes are controlled because otherwise they cannot do their business. So they spend a lot of money, and I can say for this small region in the southwestern part of Germany, it take around 3 to 4 million euro every day, and you have to every year, and you have to do this every year. Yeah, otherwise the mosquitoes will come back, and this is the way how mosquito control is performed in Germany. And it is not allowed to do to use adulticides. No fogging. This is not allowed in Germany. I think only in a emergency state, for example, if there's a severe dengue virus outbreak, I think then it is possible.
Thank you, Dr. Schmidt-Chanasit, for your talk. Very nice one. I want to congratulate you and Bernhard Nocht Institute for the effort you have put into diagnosis, the continuing effort that you have made since many, many years ago, and now with this crisis that with the outbreak of Ebola in Africa. Just a comment and then a very brief question. For those who do not know, the Bernhard Nocht Institute for Tropical Medicine is the leading tropical medicine institution in Germany. Is the oldest one? I guess is the oldest one.
Yes.
Yes, and is the biggest, and is the leading force in the tropical medicine in the continent, together with some other institutes in some other countries in Europe. But for Germany, the Bernhard Nocht Institute is the leading force, as well as, for example, some other institutes in Berlin are the leading force for the internal national infectious diseases research and surveillance. He has the best or one of the best diagnosis facilities, and for example, I know that has the largest and the best tropical medicine library in Europe, which is also a very important thing to mention. Well, but now with this background and talking about your presentation, I was very interested in this Zika situation in the transmission. I'm not too well informed about Zika, but it was noticing that it was documented the first sexual transmission case in the United States. Was it that?
It occurred in the United States, but it was the first documented transmission of an Arbovirus, sexual transmission of an Arbovirus.
Yeah. My question is, it was truly, it was only detected in semen or in saliva or in some other fluids, the virus?
Yes, I think I'm not so much. I think in semen for sure, and I think he also semen and blood, because he was diagnosed in saliva. I'm not sure, but in semen for sure. I have to look into the publication, but in semen for sure. I don't know about the other fluids now. Yeah, no, but I think in semen only. I don't know semen.
In my second,
I'm not sure. Sorry.
Okay, no problem. Yeah. My second comment in that regard is, what are the near future of the Ebola and such hemorrhagic fevers in Africa, regarding the possibility of spreading everywhere? Is the fear of everything?
I think this is not only true for Ebola. I mean for all zoonotic pathogens. Yeah, we go more and more deep into the forest. We start to do plantation there, all palm trees. It's the same problem in Southeast Asia. This high mobility. 30 years ago, it was not possible to go by bus from I don't know, from Freetown to Conakry or whatever. Nowadays, it's possible. Yeah, so this is the main problem, and that there is no border anymore between these wild animals and humans. Yeah, the border is gone, and this is the main problem. Yeah, so the distance is smaller. The distance, even there is maybe still a border. Maybe it's just one tree, but the distance is smaller. So mosquito or other insects can come to you, transmit something, even if it is not the vertebrate host itself, but this is the main problem. And globalization, international trade, not so much the climatic conditions. This is not the main problem.
I see. Together with the new lessons learned from the immunology and from the virology, remains all lessons from this old wisdom that I remember from my professors long time ago regarding infectious diseases. Infections go everywhere, people go.
Yes.
And that remains the same.
Yes.
It will be the main problem in the years
The main
to come.
problems are the humans, yes.
Thank you, Doctor.
What probabilities do you see of the Zika outbreak in Brazil to spread in the rest of the South America and coming Central America and North America?
I think everybody understands what I think about predictions. So be very careful. What I learned in the past that this ecological, that we don't understand many things about these ecological things, and we need to understand much more to say something about. What about the interference? So it used the same vector, for example, Zika as dengue. So if a mosquito is infected with dengue, it can be also infected with Zika. Nobody knows this. So there's a lot of basic research experiments necessary or need to be performed to understand more. Also the climatic conditions. What is the extrinsic incubation period? The same way like dengue, yeah? This is what we have to learn. I mean, this is the same question for chikungunya. It just stopped. It not spread rapidly in Brazil. So maybe there's another alpha virus that we so far don't know that is already in the mosquito or caused some ground level immunity in the population in Brazil. Maybe it's Mayaro. Who knows? I don't know it. Nobody knows. But we just observed this. Yeah, it came into Brazil. Everybody was concerned. Now there will be a large chikungunya outbreak, but it is so far not. Maybe during the Olympics, it's waiting for the Olympic Games. This is what we're thinking. It wait. Zika chikungunya. It will come in August 2016. It's just waiting for the Olympics.
One more question.
Thank you. Here.
Okay.
Here. Thank you. My question is there any possibility for Arbovirus causing disease to become a persistent disease? I mean, like Ebola virus to become a persistent disease, or there is no chance?
Most of all Arbovirus infection or Ebola virus infection are acute self-limiting. So self-limiting in this way that you recover or that you are dead. This is the normal way. So there are some, I mean, like the alpha viruses that cause polyarthralgia, like Ross River virus, which is endemic in Australia. So you are sometimes able to detect at least RNA in the joints. Yeah. But this is an interesting immunological feature, yeah? How this virus replicates in a low level, yeah? Not all of it in your body, but in some parts of your body that are not picked by the immune system. Yeah? So this is an interesting question. However, the normal way of Arbovirus infection and Ebola virus these are acute infections, yeah, that will lead to death or survival. And then you have immunity, and there's a restitutio ad integrum, we say. So a complete recovery, normally, mostly.
Any other question? Well, I'll make a closing remark. Early, at the beginning of the Ebola outbreak last year, projections were made about 40 million deaths worldwide, and yet it was the ample cooperations of very many countries that made possible to diminish the impact of this disease. Let's hope that we will be able to do the same in the future when any other epidemic comes knocking on our door. And I'll thank you, Dr. Jonas Schmidt-Chanasit, at this time.
Thank you, Jonas. Please. Yes, thank you, Dr. Gerardo Velasco. He was also thinking about influenza when he said what he just said about to be prepared, to try to contain it, try to really manage this infections, or these outbreaks of zoonotic infections that may come in the near future. I mean, the thing with the influenza is, as I say, not to see if it will be coming an outbreak of influenza, but when it's coming. The time has arrived to close this seminar, and thank again Dr. Schmidt-Chanasit. I would like to present to him this, of course, this letter of thanking you, this your seminar and your presentation, and I would like to present to you also this pin from our institute. You don't have a jacket, so I cannot put it into you, yes, but this is the pin of our institute. And we give it to you proudly. We like it very much to wear it with us all the time. So we hope that you can also do that this almost all the time. Now I don't have it. I have it in my coat. So thank you very much for your time coming here, and I hope that you're very successful helping also Brazil and collaborating with the Brazilian colleagues to make these Olympic Games wonderful ones. And for our work, we have started already. We hope that we can continue this work not only with the CIDICS, but also with the Institute of Public Health in Tapachula, where Dr. Ildefonso Fernandez, who is listening to us right now, is also working hardly with the Dr. Schmidt-Chanasit. I would like to thank you, all of you again, to thank the Direction for Distance Education, Mr. Rodriguez, who has made possible that everything happens. Thank you very much. Please say thanks to anyone, and also to the Direction of Informatics, who has made possible the internet broadcasting of this seminar. And Dr. Fernandez also in Tapachula, and all the colleagues that may have been following this in Germany and in Brazil. Thank you to all. Till the next seminar next Wednesday. Bye.